Hello and welcome to today's webinar on stem cell modeling for autism featuring Patricia Braga. I'm Claire Cameron, Engagement Editor at Spectrum for Autism Research News and Analysis. We'll begin the presentation in a moment, but first, know that we'll be fielding questions at the end of today's presentation, but you can ask them at any point during the session. To ask a question, submit them via the chat window to the left of your screen. And as always, I'll note that if there are any members of the press tuning in, you can only report information presented during this webinar if that material has already been published elsewhere or if you have first obtained express written permission from the presenter. Patricia Braga joins us today from Sao Paulo, Brazil, where she is professor at the Universidade de Sao Paulo. Prior to joining the faculty at the university in the early 2000s, Braga completed her bachelor's degree at the University de Santo Amaral before going on to study for her master's and doctorate at the Universidade Federal de Sao Paulo. Braga's lab investigates the underlying biology of neurodevelopmental conditions, including autism, using both 2D and 3D stem cell models. These models are created using induced pluripotent stem cells, which have been harvested from individuals with autism. These models can help us identify differences at the cellular level in people with autism as compared to typical individuals. Using such models, Braga's team has recently investigated how neuronal and astrocyte function differs in people with autism. Welcome, Professor Braga.
Well, thank you, Claire, for a nice introduction. First, I'd like to thank you all for waiting to watch this webinar. And thank you, Spectrum, for organizing this webinar. It's a pleasure to be part of the Spectrum webinar series, where I particularly found very instructive. Well, in this webinar, I will talk about the work I'm conducting at my lab in Brazil, at the University of São Paulo, using stem cells as in a dish models to study autism. Well, for some of you that are listening, maybe I'll say something that you already know, but since this will be recorded and can be watched for people not in this research area, I decided to be as much as didactic I can. So my lab is focused on disorders that affect the central nervous system, especially affecting the neurodevelopment. And in this field, autism spectrum disorder is one of the most relevant focus of study. Individuals under autism umbrella present deficits basically in social skills, including communication, and present alterations in behavior, including stereotypical movements. Well, the causes of autism are reported as genetic, with more than 1,000 genes already described as implicated, and also environmental with many possibilities, including insults during pregnancy, which is our particular interest. I would say that my lab is interested in both. I strongly believe that genetic is the base for autism, even if we don't have a candidate gene identified yet. So maybe the environment and a predisposition in genetics can both together be combined to evidence the autism phenotype in an individual. Well, since autism is a disturb that mainly affects the social brain, the basis for understanding it should be the brain functioning. So to understand the brain functioning, we have some ways to do that. So we have the postmortem brain. First, we have the postmortem brain, which is very useful, but since we have this tissue dead and fixed, we cannot study the metabolism or the communication between the cells, etc. Besides, when the organ comes to our hands, usually we have the end of the disease, so the terminal phase of some disease. So we miss the intermediate steps. So if we want to study a brain under activity, we could use an animal model, which is also very useful. Here, I should remember that an animal has his own genetic background. So the complexity of the genetic of autism spectrum disorder is not combined with this genetic animal, because the genetic background is completely different, even if we use transgenic mice. Another important thing to consider is the complexity of the brain, of a human brain and an animal brain. Here we have an example of a human brain and also a mice brain, which is just looking at them, we can notice that they are really different. So the best model, or let's say a model, but maybe the best model would say that it's going to use the human brain also with the alive cells, so cells in human brain, but alive cells that can give us some clues about the functioning of the cells. And also it will be very important to consider the human genetic background, especially for autism. So these three combined together would be the ideal models to study autism, but without brain picking. So is there any way to do that without brain picking? Well, we have recently in 2006, we have a new manuscript that came from research from Japan, Shinya Manaka. He investigated a way to produce pluripotent stem cells from somatic cells, and he used a virus, a recombinant virus carrying some genes that are expressed during the embryonic phase. And using this virus, he could prove that using that tool, we can reprogram the cells and make induced pluripotent stem cells from somatic cells. So in the case of Yamanaka, he used a skin biopsy from first from a mouse in 2006 and 2007 from human. So he did a biopsy, a skin biopsy, and using this virus, he could reprogram the cells making iPS stem cells, which are able to produce any kind of cells from our body, since we are all derived from pluripotent stem cells during embryogenesis. So in our case, we are particularly interested in producing cells from the CNS, the central nervous system, including the 2D and the 3D models. Using that, we can investigate autism to understand the basis of autism spectrum disorder, and also we can use drugs to see if we can find any drug that can be potentially used for to help the treatment, the current treatment for autism spectrum disorder. So we can call our model of study as ASD in a dish. So we can use, like I said, someone with autism, and we can collect the cells, we can reprogram the cells and make the iPS, and then from these iPS we can produce the cells from the cells that are in the brain, as the brain is our focus on this study, right? So from the iPS we can produce the neuroprogenitor cells, the first step to produce cells from the brain, mature cells from the brain like neurons and glial cells. And with these two, we can study the cellular phenotypes of the cells. Also, we can do molecular analysis, and we can verify the behavior of the cells together, studying connectivity, like synapses and other stuff. And also we can see how the cells communicate with each other, studying circuitry. Using that model, we can test drugs, even new drugs or drugs that are already in the market, like doing drug repurposing. So which is, our lab is focused on both, but I'm particularly interested in using repurposing drugs, since they are already used for other diseases. And if you want to use them, it will be faster than starting to test a new drug in terms of toxical analysis and etc., until they can be used or they can be approved to be used in human. Well, in my lab, instead of using fibroblasts like Yamanaka or other cells, I started to think about using some cells that cannot, to collect the cells, we don't leave the children to be in any pain or any issue or any stress. So I started to think about using the endoplop stem cells to make iPS, since the tooth naturally falls and usually goes to nowhere or to the tooth fairy. So because of that, first I did a trial, a pilot, and then I collected cells. I started in 2008 collecting the cells, and then you can see here a beautiful picture of the cells after we established the cell culture. So they are with this fibroblast morphology. And after using Yamanaka factors, which I make a joke here like a reprogramming machine, we have the iPS, which we can see here the beautiful morphology of a colony with these very small cells, rounded small cells. And we can notice that they are really different from the beginning. So after we published this paper, and I was feeling very confident to use them to study autism, I started a project in Brazil called the Tooth Fairy Project in order to receive as many teeth as I could in order to make a ASD stem cell bank to improve the research on autism field. So today, since 2010, this project became a NGO. So we have some people helping us to make this NGO. This is the first NGO just focused on autism research in Brazil. We just do research. We have a website and we have a page on the Facebook and emails to have some kind of communication with parents and other people interested in autism. And I'm very happy to say that we received teeth from the whole parts of Brazil. So the whole country sent teeth to us, and we have now established more or less 300 of ASD cell lineages and controls in our labs. And of course, we have some teeth that we cannot make the cell culture because sometimes they come to our lab not in a good state to do the cell culture, like with contaminations, like with bacteria or etc. So some of them we cannot establish the cell culture, but many of them are now at our labs frozen. They are frozen at cells, and we'd like to share with the autism community to improve the research on autism spectrum disorder. So I'm going to present the first results on the using the Tooth Fairy project to study autism, to model autism. It was published this year on April, and to do that we used three children diagnosed with severe autism. All of them were non-verbal, they were cognitively impaired, presented stereotypic behavior, they were non-aggressive, and at that time they didn't have any seizures. But since this work started 6 years ago, now we know that one of these patients has presented seizures. He's presented seizures now. Well, all of them were also genetically tested. We did the whole genome sequencing on these patients, also on their parents. So we did the trio, and none of them had any gene related with autism already described. Just one of them had one gene that was already described, not the mutation that the guy had. But for the others, we didn't found any genes already described as autism related. So my student Fabielle Russo did this work during her PhD. He's still working on that as a postdoc at my lab. And well, first we characterized the dental pulp stem cells. As you can see here, they were vimentine and they were CD105 and CD73 positive cells. And I'm not going to go, and I'm not going to explain all the few details here because this is published, like I said. And so after the characterization, we started to reprogram the cells using Sendai virus carrying the genes that are published or studied by Yamanaka. So after the cell reprogramming, we could notice that the cells started to produce beautiful colonies like we can see here. And then we send the cells to be analyzed genetically, because usually during these reprogramming steps, we can have some cells that present unemployed chromosomes, so we didn't want to use them. So we have to be sure that we are just using normal cells, I mean with the normal karyotype. After that, we did the characterization, the cellular characterization, and also the molecular characterization. We have some nice iPS colonies here, showing markers for pluripotency like Lin28, SOX2, and also NANOG, OCT4, and we have also the characterization before the reprogramming here and after the reprogramming, showing that the cells are not presenting mesenchymal markers anymore because they were not more stem cells from the dental pulp. They were not, now they are induced, they were induced to pluripotency stem cells. After we have the cells ready, we started to produce the neuroprogenitor stem cells. We have a very very good protocol to do that, which is also published and used by other groups. So we have here the steps that we do to until we have the neuroprogenitor stem cells. After that, we characterize the cells, and we can see that the cells are SOX2 positive, Musashi1 positive, and nestin positive, and they were negative for other cell markers that we considered as cell contaminants of the cell culture, like MG2 or MAP2. So these NPCs were very good, very nice, with a very nice morphology, and then we go over using the cells to produce the neurons. So the production of the neurons, also we have a very well-established protocol to do that. And after that, we also characterize the cells to start our investigation related to the autism phenotype. So as you can see here, we have in this slide, we have the neurons marked on green, and in red we have astrocytes, and the blue is the nuclei of the cells. And using that, we also used another antibodies to mark the synapses. And here we can have this immunofluorescence with the neurons in white. And we have two bottoms. One is green and the other one is red, which both of them, they represent the synaptic buttons of the neurons. So we quantify the synapse number of these neurons, considering the length of the branches and the puncta on the branches. So we have both the presynaptic with synapsin 1, we have the postsynaptic, which is Homer 1, and we have both together as we call the puncta, which represents the functional synapses. So we quantify all of them. And after that, we could notice that, as you can see here, the number of synaptic events from the control or neurotypical individuals and from the autistic individuals are much different. So autistic cells, the neurons derived from autistic individuals, they present less synaptic events than the control ones. Even considering the pre or the post-synaptic, both at the molecular or the protein level, we have these differences in the quantification. This means that the autistic neurons, they present less synapses than the control ones. After that, we started to investigate using the multi-electrode array. We'd like to investigate the functionality of these neurons. And using this machine, where we have a lot of electrodes here, we plate the neurons on these plates, and then this started to record the electrical activity of these neurons, and we can plot this in a graph and see the spike number, and we can notice that the number of spikes from an autistic patient compared with the control or neurotypical ones are very different. Here is just a graph representation of this graphic, so we can see using this approach that the neurons are less functional in autistic patients. After that, we did the glutamate measurement. Since glutamate is a neurotransmitter, very important to the neuronal functionality, and also again we noticed that the concentration of glutamate over time, even if in the time zero we have the same glutamate concentration, over time the neurons from the autistic cell lines, they produce less glutamate than the controls. So it reviews or it is also contributing with this less functioning of the neurons from the autistic individuals. Well, after that, we'd like to study, of course, we don't have just neurons in our brains, right? So we have glial cells. So I was particularly interested in in a glial cell that is really abundant in the brain, and also it's responsible for the connectivity between the blood and the neurons, and gives support to the neurons, feed the neurons, and is the first one to receive insults or informations from the whole body or the environment, right? So these cells are here in green. They are called astrocytes, and we'd like to produce astrocytes in vitro to study astrocytes as well, and especially the interplay between the astrocytes and the neurons in autism condition. Well, there are many protocols to produce astrocytes. We decided to use a protocol that was at the time being developed by Beatriz Freitas during her postdoc. She produced a very nice protocol from neuroprogenitor stem cells until astrocytes, and my student Fabi, she used the same protocol to produce our astrocytes from our autistic patients. And then we have this beautiful picture of very beautiful astrocytes here in this right field. We did the astrocytes characterization, and they were expressing markers for astrocytes like JFIP and also S100 beta. And after the characterization, they were screened by metabolism and also functionality. So we saw that the cells were producing relative oxygen species in more than the control individuals, which means that there's something related to the clearance of oxygen species is not working very well, like in neurotypical individuals. And after that, we did a measurement on the glutamate again, and we saw that the glutamate, even if we don't have here significance. And we can notice that there's a little tendency of more production of glutamate in astrocytes from autistic patients, which together with the neuronal measurements of glutamate, we can notice that there is an imbalance between the glutamine cycle between astrocytes and neurons. And after that, since astrocytes are also cells that are able to produce cytokines, we'd like to see the cytokines profile of the cells comparing autistic and control individuals. And we have many data on that, but we focused on IL-6 because IL-6 was already described as important to autism. So we noticed that these patients, the autistic individuals, they produced more IL-6 than the control ones, and which is very interesting, and we just keep this in our minds. And after that, we decided to do a Coculture experiment to see to investigate the interplay between the neurons and astrocytes. So to do that, we did this in different conditions. We mixed the cells. So we produced from the NPCs, we produced a mixed neuronal population, like I showed you before, that we have neurons and also we have astrocytes. And because of that, we sorted the neurons. We use magnetical sorting. We sorted the neurons, and we have a pure population of neurons. And then we plated them on top of astrocytes produced using the other protocol that I showed before, to produce just astrocytes, and then we plated our neurons on top of these astrocytes. And then we have this kind of beautiful picture, and we can see here astrocytes in green on the back, and beautiful astrocytes in red. It's very easy to find the morphology of the astrocyte, of the neuron, sorry. The size of the body, the number of branches, we can characterize morphologically this neuron, and this is very useful to understand the pathophysiology of the autistic condition. Well, doing this mixing population, this mixing experiment with these populations, we have the neurons from the controls and the ASC from the astrocytes from the controls. Astrocytes are not represented in this picture. They are in black. So we just highlighted the neurons to see the morphology very well. And so we have the neurons are this one in red. And when we have the neurons and also the astrocytes from the controls, we have this beautiful branched neuron here, as we can see on the top left. And when we have neurons and also astrocytes from derived from autistic individuals, we have this very much more simple, with the simple morphology, this neuron with less branches than the ones of the neuron observed from the control. And also we can notice that this kind of neuron presented like a bipolar morphology. And well, when we used astrocyte from autistic individual and we plate that really with lots of branches, this neuron, the control neuron, on top of these astrocytes from the autistic individuals, the morphology changes completely. So we have this neuron with less branches. Also, this neuron looks like the one from the autistic individual, and like a bipolar morphology again. And after that, we decided to do to plate this astrocyte, this neuron from autistic individual on top of the astrocyte of a control individual, and then we have the rescue, almost the completely rescue of the morphology of the neuron. So this means the astrocytes maybe are involved, very, very deeply involved in the autistic phenotype of the neurons, and maybe on the clinical phenotype of autism spectrum disorder. As I said, this work is published, but at that the time Beatriz Freitas, the girl who was doing a protocol on astrocytes, he was working with Hatch syndrome, and she had the same results using Hatch syndrome individuals. But this work is not published yet. Not only the morphology, but we'd like to see if we have any changes in the number of synapses, like we saw that the neurons from the autistic individuals had less synapses. We decided to see if with these neurons from controls individuals, if we have improvement on the synaptic number. So let's just have a look in this last graph, which is the puncta number. The first slide graph is considered just the pre and the post synaptic number. So this is a combination of the first one. Here we can see that the neurons, when we plate the neurons on top of astrocytes from autistic cell lines, we have a decrease in the number of synapses. And on the other hand, when we used the individuals, the neurons from astrocytes plated on top of control neurons, we have an improvement in the number of the synapses. So not only the morphology, but also the functionality of the neurons was improved using astrocytes from control individuals. But to me, thinking about a cellular therapy, I mean, using astrocytes to transplant astrocytes in others people had, I think it's a very dramatic situation. So I rather, instead of thinking about that, I rather think about what this astrocytes from a control individual is producing that astrocytes from an ASD individual is not producing, to see if we can find any kind of treatment instead of thinking about a cell transplantation. Thinking that in mind, I decided to investigate a little bit more about the IL-6 because the IL-6 was increased, like I showed you before. So I did we did a cell culture, a neuronal differentiation again, and then we used an anti-IL-6, an antibody to block the IL-6. And when we did that, we noticed that the number of synapses in the neurons from ASD individuals was improved. So just blocking the IL-6, the number of synapses improved a lot. And of course, we did the proof of concept. We used just the IL-6, recombinant IL-6 to treat the neurons from our control cell lines. And we have a decrease in the number of synaptogenic events. So this means that IL-6 is directly involved in neuronal functionality. So maybe IL-6, we use the principle that IL-6, anti-IL-6 could help in the treatment of autistic individuals. But until it gets on the market, it took a little bit because IL-6 is anti-IL-6 is an antibody, and usually antibodies are difficult to cross the blood-brain barrier. So this should be improved to cross the blood-brain barrier to be used for autistic individuals as a treatment. Well, not only, since my lab is working with other diseases that affect the neurodevelopment of the brain, so I decided to investigate the the effects of the Zika virus on the congenital Zika syndrome, especially because I am in Brazil, and we had at the time in 2015, we have a lot of cases of microcephaly that were born here in Brazil, especially in the northern part of the Brazil. And then I decided to use our models to see what Zika virus could do in the brain of children infected during pregnancy. Zika virus were first isolated in Africa, in Uganda, in a forest called Zika. That's why the name of the virus. And then the virus circulates a lot and until in 2014 it came on in Brazil and started to affect especially pregnant women. Well, until Zika came to Brazil, we just knew that the virus was able to infect a mosquito, and by a mosquito bite carrying the virus, one could develop this skin rash, this fever with the skin rash, like we have this in this image. But later we discovered that the virus could infect, also the mosquito could, infected by the virus, could bite a pregnant woman, and the virus could be transmitted to the fetus and cause a lot of malformations, especially in the brain, like microcephaly. And because of that, we defined that the virus was able to cause a congenital, called the congenital Zika syndrome, which we have many phenotypes, clinical phenotypes involved on that, but especially malformations in the brain. Well, we used our models, in vitro models to investigate the Zika effects in the brain, as I showed before, and we used both 2D and 3D models in this work. This work is already published. It was published in 2016. These are the 3 students that did the work in my lab. They were under PhD. One of them, the first one was a PhD student, and the other 2 were postdocs at that time. When we use it, we use it as a 2D model. We use the NPCs, the neuroprogenitor cells, and we also produced the neurons in 2D to infect with the virus. And we also use the 3D model called the Neurospheres, which are neuroprogenitor cells in 3D. And we produced also brain organoids using the Lancaster method, and also infected with the virus. Important to say that we use 3 different strains of flavivirus. We use a Zika African strain, we use the Zika Brazilian strain, and we use another flavivirus called the yellow fever, which is the one responsible for the yellow fever, just as a control to see what the effects of the virus on CNS cells. Well, first we could notice that the virus was able to infect both NPCs, neuroprogenitor stem cells, and also neurons, and the virus was able to replicate inside vesicles like other flavivirus, like we can see here. This is a mature particle of the Zika virus. After the infection, the NPCs started to come into apoptosis and also to necrosis, so they start to die. And interestingly, when we infected the neurons, although the neurons were infected, they didn't die as, we didn't have any significant differences between the control not infected and the infected ones. Maybe this is something related with the cell cycle. When we used the virus to infect our NPCs in 3D models, like the Neurospheres, like I said before, we noticed that the virus, especially the Brazilian strain, caused a massive decrease in the size of the Neurospheres. Important to say that in the first 24 hours, all of them had the same sizes, and after 96 hours, we could notice that incredible decrease, especially in the ones infected by the Brazilian strain. And we let the neurospheres to adhere to see if the cells were affected in terms of cell migration, which is important to during neurodevelopment to make the other layers of the brain. And of course, when the neurospheres were infected by Zika, the cell migration is really affected. So we have this the decreasing the size of the neurospheres and also a decreasing the ability to migrate. So both of them together, these results could corroborate to the microcephaly, the phenotype that we saw in the newborns. When we used our brain organoids, here we have a beautiful image of our brain organoids with many layers, like we can see here, characterized by some specific antibodies, which I'm not going to explain. When we infected our brain organoids, we have organoids from human and also from non-human primates, from chimps at that time. And we used both to infect with the 3 strains of virus that I said before. And in this case, we use a smaller MOI. You use it 0.1 MOI, which means less than 1 virus per cell, because we'd like to see small differences between these cells, between these infected cells in these organoids, infected and not infected. Looking at this panel, if we just go really fast to the first line and to the second line, we can notice that we have less cells in the second one, which means the cells are being infected and they started to die. So especially the progenitor ones, the most progenitor ones were especially infected by the Brazilian Zika. And also the cells that are doing mitosis, they are especially infected by the Brazilian strain as well. So after 96 hours of infection, we can notice that this incredible decrease in the cortical plate, in the cortical layer. So it means that the Zika goes to this cortical layer also and causes a massive death in the cells that are presented in this layer. And inducing the cells under apoptosis, the number of the cells under apoptosis increases a lot after 96 hours. Interestingly, we noticed that the size of the nuclei of the cells increases a lot after 96 hours, as we can see in this graph. And this is another way to measure the number of cells under apoptosis. And interestingly, when we used the organoids from the chimps, the opposite effect was observed. So in this, using the organoids from the chimps, the African strain was more aggressive than the Brazilian strain. So this means that probably the virus has suffers humanizing changes that make the virus more aggressive to human now than it was in the first time that the virus was isolated, which is something that we can expect from virus that since we have more than 16 years, the virus are changing. So these mutations make this virus now more, the Brazilian strain, more aggressive to humans than it was in the beginning. We also used animal models to see if the virus was able to cross the placenta. We used 2 mouse lineages, the C57BL6 and also the SJL. We infected both with the same Zika virus strain, with the Brazilian strain during the pregnancy, and we noticed that just the HAL was susceptible to Zika. The BL6 was resistant. We didn't find any virus in the pups, but for the SJL, not only the virus was able to cross the placenta, but the virus infects the pups, causing a lot of morphological changes in these pups, including microcephaly. The size of the head was really decreases a lot comparing with the control ones, the size of the brain, as we can compare here. And when we try to screen where the virus goes, when the virus crosses the placental membrane, we notice that the virus goes to many organs, but especially the virus goes to the brain, which means the virus is neurotropic. After that, we did a histological analysis on the pups to see the effects of the virus, and like we saw in vitro experiments, the size of the nuclei after infection, the size of the nuclei of the cells infected in the brain increases a lot. So this is interesting, but this was seen before by Bell in 1971, but at the time Bell used the African strain, and he infected the brain of the pups by an intracranial injection, and we have the same effect, but with the virus crossing the placental membrane and infecting the brain. When we started to investigate a little bit more about what the mechanisms are involved in the brain pathology of the pups, we could notice that the mechanisms were mostly related with apoptosis and also with autophagy. Coming back to the cell models, to human cell models, we also did a proteomic analysis, and we noticed that the Zika, the Brazilian strain, causes a lot of impairment in the neurogenesis processes, and also other processes related to the congenital Zika syndrome, but especially the neurological processes are crushed by the presence of the Zika. And we have many steps involved that we still have to study a little bit more, but we noticed that the synapses, especially the transport of the synapses vesicles, were impaired. After that, we started to see if we have any differences during the if we infected a progenitor cell, if you have differences in the synaptogenesis process. And yes, we have synapses impairment in the number of synapses produced during synaptogenesis after the cells are infected by Zika virus. And then we decided to investigate astrocytes. So some of this work are not published, so I will try to explain a little bit more, but I'm not going to explain all of the details because we are submitting this work now in a few days. So the astrocytes were produced, and my student Chris is conducting this work. So she produced the astrocytes and infected with the Brazilian Zika using a very low MOI, and then we analyzed the astrocytes and also the supernatant of the astrocytes culture. So she produced the astrocytes and characterized the astrocytes like I showed you before. And then we noticed that the astrocytes are susceptible to infection, to Zika infection. And after infection, the astrocytes started to die under apoptosis like neuroprogenitor stem cells, even if they are not under mitosis, they suffer more than the neurons. Comparing neurons with astrocytes suffers more after Zika infection. And we also measure some cytokines, and these are two cytokines that are usually increased after any virus infection, but indeed IL-6 was also increased after Zika virus infection. So considering the neurogenesis, the number of synapses impaired, and also the IL-6 increased profile, we started we remember the work that we did just with autism cell lines, and then we started to think if we could see any correlation between people that was born with congenital Zika syndrome and autistic individuals. So considering this findings, we know that the children are growing. They are around 3 years old. So we contact a neuropediatrician, which is Vanessa is someone who described for the first time the microcephaly caused by Zika virus in Brazil. She works in a hospital in Recife, which is the focus of the outbreak of microcephaly. So she diagnosed around 4% of children born with CZS that are also under autism spectrum disorder phenotype. So this means that probably the environmental factors and also the neuroinflammation are something that are related with autism. But of course, not all of the children who was born with congenital Zika syndrome are going to develop autism, right? And we know that because the genetic background makes difference. This was also shown by another team in Brazil that investigated discordant cases of congenital Zika syndrome in twins, and especially the twins that are not the dizygotic twins. They some of them are developed a congenital Zika syndrome, and some of them didn't. So this is related with the genetic background of the host. Well, to summarize, I'd like to leave some take-home messages. The neurons from autistic individuals presented less synapses than the controls. The neurons from ASD individuals were less functional than the controls. Astrocytes are implicated in autism spectrum disorder, causing neuroinflammation. And the Zika virus is responsible by congenital Zika syndrome in newborns. Zika virus infects and causes apoptosis, autophagy, and neuroinflammation in central nervous system, but not encephalitis as other flavivirus like the Japanese encephalitis virus and the West Nile, for example. And last, Zika virus children cohort was diagnosed with autism, which probably depends on the child's genetic background. Probably an infection during gestation triggered a neuroinflammation mechanism that could contribute to ASD clinical phenotype. This means that probably not only the Zika virus could trigger autism, but maybe other viruses infecting during this gestation could also be considered as environmental triggers for autism spectrum disorder, depending on the genetic background of the host. Well, I'd like to thank to my lab members, and I'm not going to say the name of all of them because the ones that are involved in the study, some of them I was mentioned during my talk, and also I'd like to say thank you for the founding agencies, and especially to the donors of the teeth and their families, which are very, very important, and without them we couldn't do any kind of research. Thank you.
Thank you very much, Patricia. We do have some questions, so I'll try to get through as many of them as I can. If your question is not asked due to time constraints, we have about 10 minutes, then you please know that your question will be emailed to Patricia after the webinar so that we can get that answered for you. One of the questions that we had was, have you tried using the dental cells directly from the teeth rather than inducing them into pluripotent stem cells?
This is a nice question. Of course, I many times listened the same question, and some papers showed that the dental pulp stem cells are able to produce neurons, Although the morphology, the phenotype looks like a neuron, anyone showed that these cells have any kind of electrical potential in the membranes, or they don't act like a neuron. So they look like a neuron, but they don't function as a neuron. So that's why I produced the induced pluripotent stem cells. And from this one, I produced the neurons, because from iPS, the neurons are functional. So I didn't use it neurons derived from the ectopope stem cells directly.
The next question we had was, do you think that astrocytes are perhaps more important than neurons for understanding how neuroinflammation in particular might contribute to autism?
Well, we have lots of cell types in the brain, right? So I'm a special fan of the astrocytes, but of course, they are involved. I don't have any doubt on that, but the neurons are the functional cells, especially for the social brain. So maybe the astrocytes and the neuroinflammation maybe could be a kind of root for some of the autistic individuals. I'm not thinking about the ones that already have genes involved, already described it. Maybe this is another kind of pathways for these individuals. But for the ones that don't have any genes already related, or even for the ones that the genetic background is not correlated with any other neurodevelopmental condition, maybe for these, the astrocytes could be the cell, maybe together with the microglia, maybe these two cells are really involved in the neuroinflammation and the autistic phenotype.
Just following on from that, and just looking at the results that you just presented from Zika, are astrocytes are the main focus of the investigation into the possible overlap between the two conditions?
Yeah, I have more information, but I can't present yet. But we have more information that more closely correlates Zika infection with autism phenotype. And of course, there are some, there's one paper published using animal models that also says that there are in animal models infected by Zika during gestation, the pups are under, they are with a behavior a little bit different, but it's not really a good way to compare models, animal models with people, right? I mean, I didn't use animals in this case because we have the cohort of children that was born with, the children that were born with the congenital Zika syndrome. So for these ones, I can explain, maybe I can try to explain why they developed autism and why the others are not under the spectrum, probably because of the genetic background again.
For the children who were included in the Tooth Fairy project, do all of them have a confirmed diagnosis of autism? And also, is there a plan to do genetic testing for all of the people that are included in it so that people can know genetic background for the teeth that they are using?
Yes, the answer is yes for both. Yeah, all of them are they are diagnosed by neuropediatricians, and we have this diagnosis on our hands when we receive the teeth. And of course, some of them are already genetically screened, but I don't have all of the cell lines already screened by the genetic background. I'd like to do that, and I'm looking for partners to do that. But for sure, it's going to be really important because we don't have any initiative, big initiative in Brazil to understand or to investigate the genetic background of Brazilian autistic individuals. So this is going to be very, very important, especially thinking about therapeutic opportunities because they are being developed under the American and European background, genetic background. So I would like to see if the Brazilian individuals under autism spectrum disorder, if they share the same genetic background as we can observe in other countries.
Just going back to your astrocyte cultures, we had a question which was, is it not necessary to also sort the induced astrocyte cultures because they are also a mixed population?
Good question. Using this protocol, we don't have much cell contaminants. I mean, other types of cells. That's why we didn't sort the astrocytes. We could do that because we have the opportunity to do that, but it wasn't necessary. So we have a really small quantity of contaminants on that cell culture. So we use it, we just sorted the neurons, the neuronal mixed population.
In the cell lines that you have developed, are you seeing any problems from the genetic variation between the individuals in terms of each cell line's sort of correlating results?
You mean the autistic, in the first work, right? With the autistic cell lines, right? Mm-hmm. Yeah, they didn't share any genetic background, or they didn't have any relationship between them. I mean, they were completely apart individuals, and the genes related, this is published. We have a table with the genes that we found. Just one of them had a gene that was already described for autism, but not the mutation that the guy had. And for the other individuals, they have other mutations, but none of them are was related with autism before.
Also, just going back to your results from that work, has the anti-IL-6 worked for, does it only work in specific subtypes of autism, or do you see it in idiopathic autism as well?
Yeah. Maybe I didn't make it clear on the beginning. I use idiopathic autism patients, the ones that with the severe phenotype, classified as severe. So I use these ones in our work. Of course, I'm interested on studying other types of autism in my lab. We are working on that now, but this is you know, I believe that IL-6 maybe is involved in other subtypes also, but I'm not sure. I didn't have this information yet.
Okay. I think that is all that we have time for, and I'd like to thank everybody again for bearing with us on the late start and keeping with us beyond 1 p.m. so that we could do the full hour. A complete replay of the webinar will be available on the Spectrum News site in a couple of days. This webinar is part of an ongoing series, so please check our site for upcoming talks. Some are up there now and some are to be announced soon. And there's also video archives of our previous webinars. In a couple of days, you'll be able to find the replay of this webinar there and also on our YouTube channel. Thanks again, Professor Braga, and thank you all for tuning in.
Thank you.